Okay, thanks. So we're going to start session 2. I'm really excited to introduce Abe. He's coming from Atomwise, and he's been doing really exceptional work with this company there. And I hope to hear what he has to say.
Thank you, Matt. Thanks, everybody, for being here today. So this is some work that, if you want more details, I want to put up the archive paper right here. And so that paper goes into some of the work that we're doing. We heard a little bit about ligand-based and fingerprint-based methods this morning, and graph convolutions and so forth. Our approach is slightly different in that we're doing structure-based deep learning for drug discovery. So one of the, this is a field with quite a lot of background, in the same way that computer vision, computer speech, people have been working on for decades, and it's really only the last couple of years that we've seen these massive improvements in accuracy. I like to say that I saw Captain Kirk talk to the computer, right? And the computer understood what he was saying. That was 50 years ago, and it was complete science fiction, and now everybody's walking around with Siri in their pocket, and so something has changed in that set of time. And so we exist also, as the saying goes, every beginning is a sequel, and so we're a sequel as well. So what I want to point out is here's some work. We take Autodoc SMINA, which is a structure-based prediction algorithm, and here we're running accuracy over the DUDE benchmark from UCSF. And so what this is showing is in DUDE there's 102 different targets, and then for each of the targets there's a set of experimentally known binders, a set of property matched decoys, and you ask the software to pick the aces out of the deck. And so what people typically report on this benchmark is the area under the curve. And here I'm showing at different thresholds how many targets does Autodoc SMINA achieve or exceed that threshold. So if your state of success is random guessing, SMINA gets better than random guessing on 96 out of the 102 targets in DUDE. And so that's a good thing. It means that there is truly signal there, and that it's an important beginning. 0.8 AUC is a level which people often report in the literature as quite good performance. And you can see that there's this big gap, right? That going from just beating random guessing to having good performance, Autodoc SMINA is achieving that only on 17 out of the 102 cases. And so there is progress that is achievable. So it's good for people who are in this audience and want to, improve the state of the art. Improvements are possible. And then something beyond 0.8, there's that of course not the best. It's interesting to think about what the best is because achieving an AUC of 1 on these tests is actually not possible. If you run 2 physical screens, 2 wet lab screens, you don't get 100% concordance. And there's been some investigations into this in inter-lab variation, for example. But something better than 0.9 is what the maximum is. I don't know what that level is. And so here I'm just showing at 0.9 or better, something approaching, getting closer to physical accuracy. And you can see that for that, Autodoc SMINA only gets better than 0.9 on 1 out of the 102 test cases. And so this is a level which is typically not reported in the literature, and that's why. Okay, so we think that there are a few reasons why. I'd like to tell you about one of them. And this is going to be something which the speech recognition and the vision recognition systems have also grappled with. And that's that it's hard to know what you're looking at. So the feature extraction, the feature detection, that's a hard problem. And so I like this motivating example, which is it used to be the case that for vision systems you'd sort of go in and like hand tweak what an edge detector was, right? Like, is this the side of my face or is it the wall behind me? And you sort of hand tweak these systems. When you do that, you can really only optimize over the last 5 examples that you can hold in your head. And around 2012, we all saw that if you replace that hand derivation of features with automatic statistical derivation of features, you suddenly got significant and important, practically usable improvements in accuracy. So here we're proposing to do the same thing for for molecular recognition. And so my favorite motivating example is hydrogen bonding, but you can think about it the same for pi bond stacking. You can think about it for cation pi interactions. You can think about it for whatever you want. So we say, okay, I want to build a system. I'm going to teach the computer how to find a hydrogen bond. I've got a donor. I've got an acceptor. Maybe I'll even go so far as to say that the strength of hydrogen bond depends on the distance between them, right? And so I have now some domain of repulsion, some domain of attraction. And that's not a simple curve, but I can code it for the software. And so progress has been made. But then I get clever and I say, well, also the strength of the bond depends on the angle, both in and out of the plane between the interactors. And now we start getting into difficulties, right? Because those are not themselves variables so much as functions, right? Because the preferred geometry depends on whether those are ether or carboxylic acids and so on. And so suddenly you get these complicated functions. And worse than that, the strength of the hydrogen bond further depends on the environment that the donors and the acceptors find themselves in. Are there stabilizing or destabilizing charges? Are there resonance effects? The strength of a hydrogen bond network is not the summation of the individual hydrogen bonds, right? And so very quickly, from sort of the simplest thing you would want to represent and build a detector for, a hydrogen bond, we find that maybe it's this 5-dimensional at best function that I'm willing to bet a shiny nickel no human being has ever written down accurately, right? And either you think it's this very high dimensional space, or maybe another way to think about it is there's 100 different hydrogen bonds in these different environments, and knowing when to trade off between thinking about it in one way versus the other way, that turns out to be quite challenging. Okay. This was also, as I said, done in machine vision, right? That knowing when to trade off between different context-sensitive edge detectors, that was a challenge. And so at this point, we sprinkled the magic pixie dust of deep neural networks all over this thing. So up at top, this is something I took from Hugo Laroche's deep learning tutorial. And you may have seen facial recognition tasks. So this is, you show pictures of people's faces. You're trying to identify which person that face was. But what you put into the system is actually just the raw pixels. And the system, autonomously, without human adjudication, learns to compose the pixels into edges, and then learns to compose the edges into higher order concepts, things like eyes and noses and so forth, and then those that compose into the faces themselves. And so our conjecture or conceit is that if we give 3-dimensional data, so at the top for images where you might have a grid of pixels, and the pixels look like red, green, and blue color channels, if at the bottom we have 3-dimensional voxels, and those voxels have oxygens and sulfurs and carbons and so forth, then the system might be able to learn biochemical features, and then out of those biochemical features, which you no longer have to have human adjudication for and are tuned over your entire data set, those we will compose into really separating binders from non-binders. So what's this look like? And Vijay talked about, mentioned the idea of human interpretability. So you can pick up a neuron and say, hey, neuron 214, what in the input maximally activates you? What do you like the most? And we've shown, and we presented this at American Chemical Society in San Diego, detectors that look like they are firing for sulfonyl, aromatic groups, single bonded carbons. We found sort of single detectors for metal groups. So here we're showing a video where that box is the part of the input which this particular neuron is firing maximally for. And you see that it's possibly not playing. Possibly still not playing. What you would be seeing, anyway. So there's just a series of molecules here. You can see that this box has been drawn around a sulfonyl group, and when you actually go and introspect the weights on that, there it goes. Okay. When you look at the weights on that neuron, you see that it is actually looking for the presence of sulfur and oxygen atoms in a particular geometric configuration. And remember that this was not taught to the system. All the system got was 3D atomic locations and whether things bound or not. And so the system decided that this was a pattern, this was some constellation of points, which was important and useful for separating actives from inactives. And it's such a reasonable constellation to look at that we have a name for it, which is sulfonyl group. And I mentioned that we also have other neurons that seem to fire for metals. And then actually, we have neurons which are deeper in the network that fire if those two neurons fire. So some notion of hierarchy or compositionality seems to be occurring within the network. So that's kind of exciting. Concretely, how do we do it? We take X-ray crystal structure. So being structural, one of the limitations of domain of applicability is that we need high quality structural information. And so we've done projects with X-ray crystal structures, with good homology modeling, which was mentioned earlier, some investigation of NMR. Anyway, so we take those and we discretize it. So we rasterize the input into, since it's 3D data, a 3D cube. In this case, it's usually a part of the binding site, and so this also allows you to specify whether you want an allosteric or an orthosteric binder. And then we build, and in the paper we use a 5x5x5 subcube, a 3D convolution that then we sort of drag over the input, and all of those weights are tied together. Because the idea is, if you see a hydrogen bond in this part of the interaction, you also make care in the same way about hydrogen bonds occurring in other places. And so we tie together those weights, and we learn a set of features that then we compose. So that's, and you can look at the archive paper for further details of the architecture of the paper. So that's the theory. Theory is fine if it works. And so now let's turn to whether it works. And we talked about DUDE. And this was the slide that I showed you before with Autodox Smina. And here's our performance. So for random guessing, we're essentially comparable to Autodox Smina, right? If there is signal there behind there. But I think the more interesting section is on the right side of this graph, where for the high levels of screening, the ones which aren't typically reported in the literature, 0.9 and better, we're getting that on over half of the benchmark. And so this is exciting and nice progress. And so are we done? And the answer is, of course, no, because I still have time in my talk. And so what we discovered is a problem, and I think it's one that we understand, and I want to make concrete here. We know that we might have some positive and negative examples. We might build a model, and that model might separate them. And so here I've done it. And now we have to make a judgment, which was, is my test useful, right? My test is approximating reality in some sense. And so you say, is it useful? Because I want to make sure that this thing will generalize. And if my training and testing data end up being very, very, very similar, it's actually not doing for me what I want it to do. It's not giving me a confidence that I can generalize and actually use this information when I go forward prospectively. In particular, if I have training and test data that looks like this, and in this case, red-green colorblind people, I apologize to you, but also the fact that you can't distinguish these, no one else can either. That's the point. Your test will report to you that you're doing fantastically, and you don't actually know that what your model has done, what you've selected your model to do, is to overfit and is going to fail when you go prospectively. And so what you really want is new data. And if your new data look like this, then you could measure your generalizability and in fact choose a better model that's more predictive and more accurate. Does this happen in practice? And the answer is yes. So this is a paper that Vijay cited earlier today. This is figure 3 of the collaboration between Stephen Kearns and Vertex. And here, this was a comparison of taking industrial data and either splitting it with random cross validations, which is often taught in undergrad, versus temporal splitting, which is if you have industrial data, then you also have the timestamps. And you can see that there's this massive spike at the right side of similarity of molecules. If you do cross-validation, much of your data ends up being things you've seen in your training data, or things which are very, very close to training data, 0.8 and better. But I want to point out that even if you're doing temporal splitting, there's this domain of positive controls, there's a domain of similar molecules, there's a domain of easily synthesizable, there's a domain of purchasable, there's a domain of pre-plated. There's a lot of similarity in that data. And so there's a question about whether your tests, even in the temporal splitting case, are actually going to be predictive of future performance. So this has been looked at before, and Vijay mentioned the maximal unbiased validation set earlier in his talk. And so here's one way of grappling with this bias concept. These guys define a bias term, which is the distance. You take an active and you look at the distance to the nearest active. You take inactives and you look at the distance to the nearest active. So I got to set up this data set, and so I made it that all the distances were comparable, except for this cluster here in the center. And the problem with having that cluster is if you just memorize the cluster, you're going to do really well, right? And so then it's going to fail on new and more diverse data. And I should mention, there's this philosophical point, which is if we're computational, our goal is to change the mind of a medicinal chemist, right? If you give them information where they say, yeah, I knew that already, or I was going to do that already, then it's sort of like a Heisenberg thing. You can't measure that you changed the system, right? And so were your contributions redundant? You have to make them either not do an experiment they wanted to do, or make them do an experiment they didn't want to do before, right? And that's what the contribution is. So in some sense, we as a field always have to be making predictions on the things we don't yet know, or the things which are unusual relative to the historic data. So there's also a philosophical point for optimization of the whole discovery process here. But to come back here, in the MUV, they say here you would get this analog bias, and so you'd think you're doing really well, so let's fix it by deleting some of those examples. And then after deletion, we've unbiased the data, and now we have more confidence in the performance as we look forward. So this is good. But, and especially good if you're doing things like physics or so on. But machine learning is now possible. You may have heard, and increasingly popular, as you can see from the attendance. And ignores a particular point, which is that definition of bias ignores the inactive distance. And a machine learning algorithm doesn't know that positives are positives and negatives are negatives. It knows that there's a class 0 and a class 1. And it can do that mapping however it wants. So the machine learning algorithm is free to memorize the clustering among the negatives instead of the clustering among the positives. And MUD doesn't handle that. So what we do is we make this bias symmetric by also measuring the inactive distance relative to the inactive active distance. So we flip that, it makes it symmetric. And so roughly you can think about this as, is my test good? Is one nearest neighbor enough to solve it? And if one nearest neighbor is enough to solve it, then probably your whiz-bang machine learning algorithm, people should just use one nearest neighbor, right? So what's your lift above that? Okay. So we come up with this measurement of bias or redundancy in the data sets. And then we went and evaluated over lots of different data sets. So these are data sets, and here's what's going to be the punchline, is that this bias that we measure is ubiquitous. It shows up in all the benchmarks that we've seen, and it is highly predictive of the performance of machine learning benchmarks, machine learning algorithms. Irrespective of what that algorithm is, irrespective of what fingerprint you're using, irrespective of what unbiasing technique has already been applied to the data. Because each of these people have tried to unbias the data already, either through cross-validation after clustering, so you put different scaffolds in different cross-validations, even after MUV itself, even after doing some kind of time split, and so on. Here, because I got other things I want to talk about, I'm just going to talk about the first one. We presented some of this at American Chemical Society, and the LaTeX is open in a different window. Ishar, my co-founder and de facto postdoc advisor, is telling me I need to write the paper. So we're writing this up. But let me show you the data for J&J, the first one of these. And if you want to follow up afterwards, happy to. So this is a data set which was also deep learning based, so it's appropriate for this audience. It was called Deep Learning as an Opportunity in Virtue Screening. And here they took Kemble, and they split actives and inactives, and then they did clustering so that the scaffolds were different. And then they partitioned the scaffolds, the cluster scaffolds, into different cross-validations. So they already tried to account for redundancy in the data with this scaffold clustering. Now the paper is unclear on which fingerprint they used, which metric they used, which cutoff was used, but you can get the data, and the analysis we're going to do here is totally post facto analysis. So in fact, I don't need to know any of these things. Here I'm reporting on they have about 1,200 targets. Here we report on 550 which are big enough to do some work that we want to do next. So here's what it looks like, and this is a complicated slide that is kind of showing up. This top one is we order the targets by how much of this bias term there is. And so you see some targets have little and some targets have a lot. Below there is AUC for 4 different machine learning algorithms: random forest, SVM, logistic regression, KNN. And you can see just by eye that as the bias goes up, the AUCs go up. We can make that concrete by plotting AUC versus bias. And here you see trend lines that go up again. And random forest, for example, has an R squared here of 7.1. Okay. Once you have this bias term, you can do sort of the same thing that the MUV paper does, which is, can we unbias in the same way by dropping some data out? And so we went and tried that. So this slide, that's this top half before. On the bottom here is the 550 unbiased. You see that we fail to unbias for about 150. Those are either small or highly clustered data sets. So if we can zoom in just on the ones where unbiasing succeeded, so the subset now of the 400 where it succeeded, you see a couple things. First of all, the correlation, as you would expect, completely disappears. But also, all of these algorithms take a massive hit in the predictive performance. So random forest, which is popular, goes from 0.83, which I think we'd be happy about, and which we'd publish, down to 0.66, which I think we'd be unhappy about. And that works same for pick your favorite, right? SVM, logistic regression, KNN, and so forth. Okay, so where are we left with? This analysis left us, kind of skeptical about the predictive performance of benchmarks. And so as Vijay said, the next step is to do prospective validation, because that's really modeling how these things are going to be used, because it's how these things are used, right? So I'd like to share with you 2 stories. The first one of prospective, these are cases where there were no known ligands, and so ligand-based methods, first of all, were not appropriate, and also where we're really doing new discovery. So nice case studies. This is Ebola. We got involved in fall 2014, really at the height of the pandemic. What we're doing is targeting, with our collaborator Jeff Lee at University of Toronto, we're targeting a particular viral fusion protein called glycoprotein 2. And glycoprotein 2 undergoes this conformational shift from an open state to this closed state on the right. And so what roughly we want to do is park something to block that conformational shift. So it's sort of intra-protein blocking, because conformational shift. And so what we're going to do is this central trimeric core, we're going to sort of pull this guy off and park something right there to see whether we can block viral fusion, viral entry. Since it's a neglected tropical disease, drug repurposing seemed appropriate. And so there's about 7,000 molecules which had phase 2 or better data. That's from US, Canada, EU, and Japan. We analyzed those. The top 17 were bought and then tested in a cell-based assay. And so here, I'm sure you guys have seen these kinds of videos before, but here you can see different medicines being different poses generated, and then each of those is scored by the 3D convolutional neural network that we talked about before. Okay, so the assay was kind of neat. Jeff set up a pseudovirus assay so that the pseudo Ebola virus would inject green fluorescent protein. And so that means that by counting the number of cells which are glowing, you can get a measure of infectivity. All right, so one compound, a couple of compounds, but one compound I want to talk about, blocked Ebola infectivity by about 80%. Now, sulfuric acid also blocks cells from glowing, and so you want to make sure that you're not just killing them. And so that same compound also passed a cell metabolism profile. So it doesn't seem to be grossly just breaking things. Because it's a phenotypic screen, I can't tell you that it's working for the right reasons against this target, but Jeff did a counter screen for a virus with a different entry mechanism, and the compound correctly failed to inhibit that viral entry. Okay, so it's some evidence that it's working something unique to Ebola. And we were able to get a cell-based IC50 of 12 micromolar. So either that could work as a lead, or if you don't want to give up the safety data, then you could try that for people. The interesting thing is that this is a real repurposing. So it wasn't the case that this was, it worked for influenza, and so let's use it for Ebola. This was a drug which actually was developed for central nervous system disease. So really very different systems, no previously described antiviral activity. It seems like a real example of repurposing. Okay, so the next story that I want to tell you about is multiple sclerosis. And so in this case, we met our collaborators sort of partway into their work, where they had shown that if you could block a protein-protein interaction, actually receptor-receptor interaction that happens on the surface of the neuron, that good things happen for their multiple sclerosis mice. And the way that they had shown that was they chopped up each of the interacting proteins into short peptide sequences, and by dosing the peptide, it would just occupy the binding site and then block interaction. They had tried to commercialize this, but ran into problems because peptides don't really cross the blood-brain barrier, and so it's acceptable to treat your MS mice by directly injecting into the brain, but it turns out that people don't really like that as a treatment option. Also, peptides sometimes have systemic immune response, and so death is a side effect, and so people don't like that as a side effect profile either, which is the sort of thing you learn when you try to go, cross the academic-industrial divide. And so here's a case where the biology was really well worked out, but there was no supporting chemistry, right? And it was an interesting case because protein-protein interaction, that's a tough target. You have to cross the blood-brain barrier, so that's a tough target. This was something where there were no known ligands. And so you're sort of out in the wilderness. And also, but the nice thing is that peptide sequence also labeled an area on the protein where we could look for a binding site. And so just by eye looking at it, we thought, hey, maybe that's amenable to small molecules. So if you're willing to try small molecules, let's try small molecules. Initially, they'd asked us whether we could do peptidomimetic design. We said, yes, just no better than anybody else at a whiteboard. So fortuitously, we were the biology worked out that there was a pocket there. Anyway, they were game, and so now the rest of the slide. So that's background. Because we don't know how to block protein interaction per se, we cast a pretty big net, and so we took what at the time was the entirety of the zinc immediately commercially available subset. And so we took those 8.2 million, and we did filters, Lipinski-like rules for oral absorption. We did Lipinski-esque rules for blood-brain barrier penetration. We filtered out pains and aggregators, autofluorescers, other false positives. And we were left with 500,000 molecules, and that's actually what we predicted against that target. You get a rank list of 500,000, and they had hired a medicinal chemist who had decades of experience in central nervous system diseases. And he looked through the top 2,000 or 3,000 by hand, which as a computational guy, that sounds amazing to me. But he looked through the top several thousand by hand and thought they looked both reasonable and interesting. So that's a good part of the Venn diagram to land in. And then he said, I've picked some molecules. I'm curious what your system thinks about my molecules. And so he gave us 22 molecules, and we ran them through. And I have to tell you that this was absolutely terrifying, because the best one was number 100,000 out of 500,000, right? And so you have to go to the guy who's the gatekeeper, right, who you want to like you, and say, these are terrible molecules and you should feel bad. And that's a hard conversation to have. So we tried to be polite and Canadian and say, statistical system, maybe it doesn't understand everything, early days, what can you do? These are terrible molecules and you should feel that. And he said, good, I gave you duds. He said, I've worked with computational systems in the past, and the false positive rate was what killed us every single time. So I wanted to see your system reject molecules before I let the project go forward. And so having rejected them, the project went forward. They bond tested the top 50. Eventually they tested another 200, but all the hits were in the top 50. 9 of those 50 were binders in a Brett assay. 3 of those were active in a follow-up in vitro functional assay. And again, I predict binding, not activity. So, whether agonism, antagonism, 3 were doing the right thing. One of those they picked to dose IP in mice and then orally in mice. We ended up with 3 nanomolar per gram. And here I think, the use of Comic Sans shows that it is not my slide, but just directly the collaborator slide. And the top should read, small molecule treatment significantly improves neurological function in EAE mice. Now I'm not a biologist, but I have a happy looking mouse and a very sad looking mouse with the dragging hind limbs and limp tail, which is indicative of multiple sclerosis symptoms in this model, and then a nice separation between the treated and untreated curves. So, of course, this is not just, not really my work, but the work of the team, many of whom are here in the audience today. So find these people and talk to them about how we do any of this stuff. And if there are questions, I'm happy to take questions.
On your graph of AUC versus bias, I was wondering if you were able to extrapolate back to a bias of 0 and get a sense of what the AUC would be like in the absence of bias.
That's a good question. So there are some subsets where there is a bias of 0, right? Just depending on how we, for some there's more bias, for some there's less bias. Mostly the way we were looking at it is, if you're going to assess, if you have a benchmark, right, you should probably care more. You could weight your belief in the outcome of the benchmark relative to how much bias there is, right? Like, it's telling you something, but is it telling you the right thing? And so it might just be a good sanity check to run over your benchmark and say, do I really, to the extent, there's Bayesian explanations, right? You get great performance on a benchmark and you say, either I'm a genius and my technique really works, or the benchmark is terrible, right? And life sucks. And so the question is, how depressive do you feel that day, right? And so this might give you some evidence about where to put your belief on that baseline. But it really depends on the details of the model and so forth. Yeah. Yeah.
This mistook this, but one of the parameters in your model seems to be the volume of the voxel that you use.
Right.
Initially, does that change with different targets, or using the same voxel volume, or the number of voxels you can use different targets?
Sure.
You comment on how that makes sense.
Sure.
Can you repeat?
Yes. So the question was, there were a couple questions. One of which was, what do we change per target? And specifically, the question was, voxel size. Do we change that per target? So we've looked at some of these things. There's a number of hyperparameters that you can tune in these from input. How exactly you parameterize the input and how you show it, to what stochastic gradient descent algorithm do you use, to what's the architecture of your network and so forth. What's the size? I showed you 5 by 5 by 5 convolutional filters, and so in particular this case was, what's the size of the volume of a voxel, right? And so the answer is, yeah, we've looked at a number of these things. I have no principled way, just like brutal empiricism is the way, and none of my machine learning friends can tell me like what the best way of analytically deriving the answers to these things. And so in some sense there's no reason to believe that these are optimal. We've tried a lot. I encourage you to try more. And we saw this actually in computer vision as well, where AlexNet had these huge improvements in performance, right? Choosing 7 by 7 low-level convolutional image filters. And then 2 years later, Fergus and Zeller did a really thorough analysis of what those, or 11 by 11, what those hyperparameters on the architecture should be, and got much better performance, and 2 years later won the challenge. So there's no reason to believe that the first one was the best one. Sort of in the same way that when we do drug discovery, we have to, you find a molecule and then you test a bunch of things to make sure that was the best one and try to find a better one. The other part of your question, which is what changes target to target. So we have this sort of ongoing process of deriving the best answer to these questions. And so there is some symlink, which is current best model, right? And you can run predictions any given day with that one. And as our assessment changes, we might use a different model and switch to the different model. But we have one global model for all targets. These are not reparameterized per target. And so that's why both in the Ebola case and the multiple sclerosis case, these were cases where there were no positive controls for us to use for these targets, right? And so these were real blank piece of paper designs, right? And so whatever, as models train, sure, that changes, but it's one best one across any target that we're doing prediction on. Yes, sir.
I'm involved in evaluating different technologies for identifying hits for projects at Denali. We can do a traditional high-throughput screening, we can do DNA encoded library screening, we can do a virtual screening.
Yeah.
Tell me why we should work with Atomwise to do a virtual screen and find molecules and test it versus a traditional HCS screen or another screen.
Sure, great question. How well do those work for you?
Generally, the HCS screens do identify hits. We don't have our own HCS infrastructure, so it's a cost, right? So you can hire a CRO to do a screen for you at $500,000 or $300,000 or whatever the cost is relative to doing a virtual screen, where the success rate is not as high as, I would say, it hasn't been as successful as doing this traditional experimental screen.
Okay, so it's a perfectly reasonable question, right? And if there exist technologies, then you should use the fastest, cheapest, simplest technology that exists for you, right? I'd say one of the advantages of using virtual screening is that we can, there's a couple things, right? Sort of in the same argument as DNA encoded libraries, probably the pitch versus traditional high throughput screening is that you can test a much broader diversity, right? And we've got the same kind of pitch here. The other one is, Brian has published papers where you show that the success rate is actually better with virtual screening after you get rid of all of the artifacts and all of the already knowns and so forth, right? So here, I think the punchline would be that the kinds of errors you get with one technique are probably decorrelated with the kinds of errors you get in another technique. And so you can get new parts, you can explore new parts of chemical space, which these other DNA encode libraries or traditional high throughput screening might not be able to thoroughly vet. The other argument, of course, is computational techniques like this one can analyze molecules which have never been built. And so with respect to going into new chemical space, and deciding whether you wish to synthesize or not, this can provide guidance in a way that existing high throughput screening or DNA can't. And so that's an important just categorical difference between the two. But my conclusion would be, let's talk.
Okay, one more question.
So in computer vision, it's not uncommon to initialize your network with the weights learned from ImageNet.
Yeah.
Does something like that exist for AtomNet?
Yes, it's called AtomNet. I mean, yeah, like there are people who are now looking at doing 3D convolutional neural networks and so forth, but I don't think that there's a standard. Barth, you've got deepchem.io, and I saw some 3D convolutions in there. Do you have weights that people can download?
It turns out to be challenging to build these networks. We have some research in that area that might be quite ready for easy download time. So maybe wait a couple of years is probably my best recommendation.
Yeah. So come talk. If you've got projects, come talk to us, I guess. All right. Thanks very much.
Great, thank you so much.